to the first Wildlife Wednesday series. Peter and I wanted to kick this off with, he is our first speaker, but we've got a lineup of a different biologist speaking once a month for January, February, March, and April. Next month, we will have a biologist from UBC talking to us about painted tortoise, Oregon frogs, and Checkerspot butterflies. She's actually involved in the project over on Hornby. You were handed a piece of paper when you walked in. It's got a QR code on it. We are partnered with Wildlife Preservation Canada. Peter has done work with them in the past, so he had that connection, of course. And if you put your camera on it, it'll send you to their website. Our Purple Martin project is actually featured in their annual report. I was hoping to have hard copies for you, but they haven't arrived in the mail yet. But you can go to their website. We are listed there so that people nationally can actually donate to our program as well. So baby steps, we're starting baby steps, but we do have great big ideas moving forward.
What I want to do is to introduce you to the genesis of this Checkerspot butterfly breeding program in Canada. But let me get back a little bit to a little bit of a history of the Checkerspot. The Checkerspot, the Taylor's Checkerspot, became extirpated, which means regionally extinct, in Canada. The last seen on Hornby in year 2000. We think the Checkerspot disappeared on Denman before that. There used to be good population actually, and all the way up the coast, Victoria, etc., 100 years ago. The animal has a need for a kind of we call it the Garry Oak habitat. It's open grassland more or less, and but certainly not deep forest. So the interesting is that when we had the pretty significant logging in the Northland, it changed the environment from a forest to a quasi meadow habitat. And the ruts that were left by the machinery created ponds. The little ponds, the seasonal ponds, was exactly what the food plant needs. The food plant is called the Veronica scutellata. The other word is marsh speedwell. Needs these spring wet ponds, and it senesces sometimes later in the year and then comes back every spring and provides incredible amount of food for the Checkerspot, which is a historic food for this particular animal. So we then were actually dismayed a little bit that this animal had disappeared. It was our very Jenny Balk who found them on the settlement land in 2005, if I'm right, and in May, when the adults were flying. And this was great news, and the Ministry of Environment got very excited about it, and biologists came out and surveyed Denman Island, and lo and behold found other population. One very interesting one is the lands owned by Mike Morrell, the Middle Farm, I guess it's called. And then, of course, they were on settlement lands, and then we found a real nice population at the entrance of the Park Road, and in the Checkerspot, I should say the butterfly reserve was in the Northland Park, a 10-hectare area set aside because it did have good population. So that was 2005. Then it went, we still got some pretty good reports by biologist Nick Page or something, I forgot the name now, who's found quite a good population, but it was dwindling. So in 2012, we already had a Parks Committee, and the Parks Committee had a number of interest people in what are we going to do about this checkerspot, because it was dwindling. And why was it dwindling? Because the habitat was changing again to a forest. And worse yet, the Scotch broom came and took over the place. So we had to do something, and obviously remove the Scotch broom and remove and cut down some of the conifers that were reoccupying the habitat. So we created a community working group. And if you have the time and you go around and you see a little easel over there are 40 names, and might not even remember all of them, who were volunteers, typical Denman response. 40 volunteers went out, and we took down broom, you wouldn't believe it. The fire department had to come and burn it, big rows of this horrible plant. And then later we actually took a lot of conifers down to just open this habitat again. And the Ministry of Environment is still maintaining that quasi-habitat to be an open, more or less meadow-like environment. Well, the other message was the butterflies still not increasing in numbers. And so with my background, having bred endangered species for reintroduction, many different species, exotic species, of course, internationally, I said, well, why don't we try to breed this animal? Can we breed this animal? I knew nothing about checkerspots in September 2012. So I Googled checkerspot conservation, and up pops Oregon Zoo breeding these animals in captivity and doing exactly what we were thinking about, releasing them in 13 different sites in Oregon. That's, again, also a very limited area where they live. So I talked to the director of the Portland Zoo, and I said, can I come down and have a look? And he said, absolutely. I was still, and still am, part of this international zoo family. We just help each other spontaneously. So there was Erica Bland and John Dillon and myself and a friend of Erica. December the 3rd, we went down to Portland and got a 2-day crash course on how to breed these things. And they showed us everything. And you will see, actually, the procedures in one of the videos. There are 2 videos. It's called Husbandry. So the amazing thing was that there was all kinds of enthusiasm on the island to do that. So I went back to Jennifer Heron, the chair of can you all hear me? Should I do this closer? The vertebrate specialist, invertebrate specialist for insects, etc. And I said, why don't we breed this animal in Canada for augmentation? She said, oh, we'd love to, but we don't have the expertise, we don't have the money, we don't have the space. And I said, I have the expertise, I have the space, and I will find the money. She said, well, really? Well, yes, just give us the okay. She said, run with it. And I have to give Jennifer big credit for being as gutsy to do that, because when you're working in the government system, which is complicated, all these different approvals you have to get and everywhere, she had the leap of faith to say, run with it. And then we sort out the policies and the agreements and memorandums of understanding later. And we did. And John Dillon was probably one of my biggest helper, and there were others, but we changed one of the well, I call it the support facilities for I was breeding endangered species of birds, soft-bill birds. And so I had a kitchen and space. So we converted that to a laboratory. You will see what we did. And by May 23rd, and we didn't get, the government had actually no plans to get any specimens to me because they had to get all this paperwork done. But by May the 13th, I see Nettie sitting here. Nettie and Mike Morrell and Jenny Balk There was also Chris Junk from the GERT organization. They walked around on Mike's middle farm land. They wanted to see a checkerspot because in May is when the adults are flying, just pretty well just that month. And they noticed a female laying eggs under a narrow-leaf plantain, Plantago lanceolata, which is another food plant, but it's not a native one. It is one that for some reason this animal likes to eat. And it likes to lay its eggs under it because it likes to lay the eggs under an overhanging leaf. It was this plant was right on a horse trail, so to speak, and the future for it was very much in question because it would get trampled. So we made an executive decision to move them without getting prior approval from the government, to move these into this laboratory we had already. Everything was ready to go. We had the lights, we had the temperature, I had the food plants already cultivated in anticipation. And so there were 18 eggs. After 18 eggs, there were 13 that were fertile. And they turned into tiny little larvae. They're about 2-3 millimeters long. So I kept feeding them and looking after them during 13. And of the 13, 12 made it all the way through all the different instars. That's what they call each time when they change the exoskeleton, to the spring. And in the spring, and then of course, if this animal has a very funny biology, it sleeps from August, September, right till February. And then it wakes up and then it starts feeding like crazy and, you know, looks for these marsh speedwell, of course. And then it gets enough body mass and it pupates and then it hatches into butterflies in May. And then they lay eggs again and then we are having the same circle again. So then those 13 butterflies that we raised from the 18 eggs were then basically the gene pool for this population in captivity. But of course, any geneticist, anybody involved in genetics and what knows, that's not ideal. They were all brothers and sisters, so we had only one genetic expression of all these animals. So I contacted Woodland Park Zoo, which is the Oregon Zoo it's in Oregon Zoo in Portland. And I said, what should we do? And she said, well, get permission to collect some females at the very end of their laying period. They pretty well, they hatch in May and they're just about done laying their eggs, and the males and females all die. But if you can get them at the tail end, they may still have some eggs and they may still lay some eggs. And sure enough, we found 6 of them and had a good number of eggs from them. So this was, of course, 2014. We were very, 2014, it was the second year of looking after them. We had a bit of a dilemma, and we did a research project by doing so to come across it. And it takes quite a bit to discuss that, but what was happening, all of a sudden some of the larvae were dying, and I'd sent samples out to very, very highly specialized institutions and research facilities, and they said, no pathogens, no fungus, no bacteria, and no parasites. And the food that you sent had no contaminants. It's husbandry. And I said, how in the heck can that be? And then I was reminded that when I was breeding greater wax moths, don't worry, that's not the one that lives out here. The greater wax moth is a typical food item for soft-billed birds. And I had the same event, and I had sent animals again at that time, or the first time, to these research facilities, and they came back, no pathogens, husbandry. Now, what was going on? What was going on was population density. Can you believe it? It was stress by too many individuals in a given space. Well, that's my conclusion. We all know that if you take 2 pairs of laboratory rats and put them in a perfect environment, They will have young ones, and they will even communally looking after each other, and it's just a joy of life. And if you don't remove any rats, they get more and more and more, and in the end, they become aggressive to each other and even cannibalize each other. So a complete change of behavior. So why should invertebrates not have that problem? But anyway, so we then did, I don't know how many tests, talked to specialists in testing them a different way. They could not establish any infectious event. So the upshot is that we changed the protocol and just reduced the number of animals. They always were fed in little cups, in little 16-ounce deli cups. Every time by about 40%, every time they change their exoskeleton, so it got bigger. So they started over with 100, and eventually in the winter when they sleep in their little buckets, there were only 15. And lo and behold, since then, never had that issue again. So yeah, I don't want to make suggest to make a cross test and do this again, but it clearly was the issue. It then went on. We'll get to the videos very quickly. So then we're in 2015 came along, and we knew now not to do that. And we raised about 2,000 these larvae and released about 120 on Denman Island in the Butterfly Park. I had a 3-year contract with the government, which was a kind of a one-sided government, and Kitchley said, "Do you think this is a contract or what is it?" And I said, "What do you mean?" He said, "Well, don't you have any business sense? There isn't a single commitment in there of funding or for this. You have to find the funding, you have to produce all the animals, you have to find the people, you have to build this, and you have to do this for 3 years and come up with 1,000 butterfly larvae." I said, "This isn't a business, this is a passion." So that's how we got around that problem. I actually see John Mills sitting here. John Mills took over the working group. So in the meantime, we kept working on the habitat in the buffalo, in the park, in the butterfly park, butterfly reserve. And the Ministry of Environment then got interested in bringing the Checkerspot back to Hornby. So after 2016, the animals were moved there, and they have in the meantime done that. So one thing that I would have to tell you was absolutely fundamental, critical, and still is the generosity and the enthusiasm of Wildlife Preservation Canada. Because when I was faced with the question, who can help us funding this? I had to find, we were looking at about $10,000 operating money, which also paid for a student for 3 months. Wildlife Preservation Canada, and I was actually befriended with Jerry Durrell, who was the founder of the Jersey Wildlife Preservation Trust in Europe. And they have this sister organization, which is Wildlife Preservation Canada. And I've worked with them, I guess, on other species in Calgary, and they enthusiastically took it on. And they still, Wildlife Preservation Canada was a single funder of the Checkerspot program. They're still doing it because in 2016 I moved the equipment and a colony of these animals to the Vancouver Zoo, because the Vancouver Zoo provided some space for Wildlife Preservation Canada to do endangered species breeding. And what adds very nicely to this talk today is next month's talk when Andrea Gielens comes. She's the lead biologist of this conservation unit at the Vancouver Zoo, and they raise for some time and return to the wild western painted turtles, Oregon spotted frogs, and then of course since 2016 the Checkerspots. So with that I think I should, well, there's one other thing that is kind of interesting. If you look into the websites, the write-ups by the government or any other organization involved with it, there is no mention of actually the fact that we on Canada, a handful of us started this, got the idea, started it, got it going, and then passed it on. It's all now, of course, credited to of course, the Ministry of Environment. And I just wanted you to know that you were really, if you weren't, I can look around the room and I can see several people who are jumped on this. If you wouldn't be there, and if I didn't have this confidence that we could do it on Denman Island, I am wondering whether this project would ever have come off the ground. It's that simple. It's like buying a puppy. Once you have it, you got to look after it, and so you can't let go. And I think it's that sort of kind of a thing. All right, Wildlife Preservation Canada, of course, is the one organization that does give credit to us, and Andrea Gielens, who got the award, got a Peter Karsten award by the Canadian Zoo Association just in October. Peter Karsten award for in situ conservation, because they managed to bring the checkerspots to Hornby in great numbers, several years in a row, and finally they have a resident population. So they have brought it back. So that was a real winner. And this was of course, Andrea's work. So you will talk to her next week. And it was kind of nice for Wildlife Preservation Canada when they said they were particularly honored to receive this award because it was named after the person who founded the project. So it comes home. So there we are.
Nowadays, we are very concerned about the disappearance of our pollinating insects, such as bees and butterflies. World conservationists say that it is not just species which are endangered, it is their homes which are in peril. To save our pollinators' habitat is our biggest challenge. The tailless checkerspot only exists in a handful of places in the entire world. Amazingly, in Canada, it only lives on Denman Island. However, it disappeared from Denman Island in the mid-90s, but after extensive logging in the northern part of the island, it was found again in 2004. It prefers open meadows and does not like to live in the mature forest. The logging machinery created deep ruts which turned into small pools. This is where marsh speedwell likes to grow. It is a preferred food plant for the checkerspot butterfly larvae. The butterfly feeds on the nectar of various flowering plants, especially wild strawberries. The sudden abundance of speedwell allowed the checkerspot population to grow into the thousands. However, the forest is reclaiming the meadow habitat, taking away sunlight and water from the food plants. Invasive plants such as Scotch broom are another real threat. Luckily, the Denman Island community rallied together to establish a 10-hectare butterfly reserve in the BC Provincial Park. Volunteers formed the tailless checkerspot community working group to remove invasive plants to preserve the meadow habitat. We are also building food gardens for the checkerspot butterfly by creating ponds with pond liners. This is where marsh speedwell grows throughout the summer. Flowering plants are relocated to the food gardens to provide nectar for the adult butterfly. The food gardens are funded by a grant from Wildlife Preservation Canada and Walmart Evergreen grants. Since the fall of the year 2013, I have been energizing this project, developing the conservation breeding program. Because of the threat of the extinction of the species, the Ministry of Environment supported the building of a tailless checkerspot conservation facility. The facility has 4 rooms. What you are looking at is the lab where all the technical work is done. The second room is a room where larvae are raised under artificial lights, which is a feeding room. And the third room is the sun room where we do the mating. And finally, the diapause room where they sleep for the fall and the winter. Checkerspot butterfly life cycle takes an entire year from butterfly to butterfly. In February, the larvae sleeps, it's called diapause, which will wake up in March and feed heavily to turn into a pupa, and then into an adult in May. Eggs will be laid late May to June, and then the larvae hatch in June and grow all the way to insta5 in August, when they turn back into the sleeping mode called diapause. By continuing working together, we shall have an opportunity to preserve this species for future generations.
It's called the husbandry, and it was done to pass on our experience and our knowledge to the Vancouver Zoo to breed these particular animals in captivity, or anybody else that wanted to breed Checkerspot. You've probably seen a young lady, and you'll see her again in this video, and there is some duplication, but I apologize. And it's Forrest Hunsinger. Where are you, Forrest? Forrest was a very young lady. She's here somewhere. There it is. And she worked with me day after day after day in feeding these butterflies, and practically grew up with the butterflies. So that's just one person who really helped. And of course, as I mentioned, John Dillon. So let's run this one. It's basically a training document for Checkerspot breeders.
The tailless checkerspot butterfly became extirpated from Canada in the mid-90s and reappeared 10 years later on Denman Island after some heavy logging removed the forest and created meadow habitat. The butterfly larvae feeds on speedwell, which grows in moist places, such as the ruts left by heavy machinery. However, forest succession is reclaiming the meadow habitat, which destroys the food plants of the butterfly. This led to the initiative to breed the species in a laboratory environment for reintroduction and augmentation in the wild. The facility has 4 rooms. What you are looking at is the lab where all the technical work is done with the fridge and all other equipment. And the second room is a room where larvae are raised under artificial lights, which is a feeding room. And the third room is the sun room where we do the mating, which is necessary with sunlight. And finally, the wintering room where the animals are in diapause. The Checkerspot butterfly life cycle takes an entire year from butterfly to butterfly. In February, the larvae sleeps, it's called diapause, which will wake up in March and feed heavily to turn into a pupa, and then into an adult in May. Eggs will be laid late May to June, and then the larvae hatch in June and grow all the way to Insta 5 in August, when they turn back into the sleeping mode called diapause again. The larvae are kept in the diapause room under clay pots to regulate humidity and temperature. A logger monitors it, and we must maintain a minimum humidity at 40%. Larvae are checked frequently to see if they are healthy. However, they are not active and immobile. We feed 3 species of plants to the larvae. This is Veronica scutellata or marsh speedwell. This is Plantago lanceolata. And this is Veronica serpilifolia or thyme leaf speedwell. Of the offered food plants, the young leaves of Veronica scutellata are the preferred food, as you can see. We prepare little bouquets of Plantago and Veronica and put these into custom-made vases, which are filled with water to keep the food fresh. These are placed into the bin where the larvae are fed and also receive brown paper, which they like to pupate, and we weigh those larvae on a daily basis and return them after weighing to the bin where the food is provided, and we also provide extra moisture by adding 6 cc of water to maintain a minimum of 40% humidity, again monitored by the logger. The bins have double screens to avoid parasitic wasps from puncturing through the screen into the larvae and to lay their eggs in it, which eventually will kill them. The larvae feed heavily until pupation, and in this case you see them in Insta 6, which is the last time they shed their skin, after which they will turn into pupae. The larvae stop feeding before pupation. They are about 3 centimeters long and hang themselves in a typical J position under the paper. You can see here also the last exoskeleton, which would be the 7th skin, attached to the point where the pupa is attached to the substrate. Every pupa is weighed and identified to maintain records of the matriline, that is their mother. The pupae are stored in plastic containers awaiting eclosure. The hatching of the butterflies falls usually into the month of May, which is called eclosure. The butterfly first must rest and expand its wings before it can take flight. This can take several minutes. All butterflies are weighed because we are able to tell the sexes by weight. Females are always heavier. The sexes are told by looking at the posterior of the butterfly. The females have an oval pattern of bristles with an open center. The males have a closed pattern called claspers. Females are stored in plastic cups with paper lining in the fridge. They are fed by hand before mating. Males are held in transportation castles, and they are fed on a daily basis by providing them with natural flowers, in this case English daisy, and also by feeding them by hand. We do that by stimulating them on flowers, like in this case the scabiosa, and then quickly offer them honey mixture, which is buckwheat and water mixture 1 to 3 from a Q-tip. Males are kept in a screened enclosure and taken into the sunlight. Females are added to the enclosure, and males will soon begin to copulate with the female. When the two animals are facing in opposite direction, copulation actually starts. It must be measured by time. We consider it successful when it is more than 50 minutes but less than 3 hours. Some wild females have been collected to add genetic diversity to the colony. These are captured in the field and bored for a short period of time. GPS recording is done, and the butterflies are put into tubs. These are called oviposition tubs, where they will lay their eggs, as you can see under the underside of a plantago plant. A female might lay as many as 50 to 100 eggs. Once we have collected enough eggs, they will be released again to the wild. They are still gravid, still able to lay more eggs. Eggs are laid on the underside of leaves. They're at first yellow. Then they will turn into a brown color, and after that, close before hatching, in a silver-gray color when the head cap comes off and the larvae hatch. Eggs which are not fertile remain yellow. The little larvae will soon wander out onto the leaves of the fruit plant together. The daily routine starts with putting on rubber gloves and collecting food plants. Veronica are grown in the garden in beds. Fresh leaves are collected from Veronica as well as plantago. They are bleached with 5% household bleach, rinsed and spun dry for feeding. The keeper is now ready to take the larvae into the lab for feeding. The utensils which are used to handle the larvae are sterilized with isopropyl alcohol. The alcohol is then rinsed off. The lid is removed and the larvae are taken out of the container and placed in a fresh container with fresh paper. Larvae here are seen basking at the top of the lid, and others are hidden inside the paper with the food plants remaining after feeding. The leftover food is removed and placed in a separate dish. It is important to look for larvae in the paper so none is overlooked. The paper then is inspected on both sides to see that no larvae are hiding anywhere. All the debris is kept and inspected later for any overlooked larvae. The larvae are picked up by sweeping them with a soft watercolor brush onto a spatula made from a plastic margarine container. Leaves of the food plant are placed loosely in the container. Another method is to use glass vases. These are helpful to keep the plants fresh, and they're then placed inside the food containers. It is very important to keep track of the genetic lines of the larvae. This is done by having the ID of it on a label, and the label must be religiously moved from one cup to the next. We have created screen bottom cups. These are inserted into a normal cup to collect the frass. The frass falls through the bottom of the screen and stays away from the larvae. As the larvae grow, they turn into different instars, which means shedding their skin and changing their shape. The shedding should progress smoothly. The head cap is the first thing that's removed, and the skin is shed just like a sock. Here the larvae is doing a dance to fluff up its bristles. We see an instar 3 and an instar 4. As they change, they get larger and have more bristles. This is an instar 4. And here we see instar 4 and instar 5, and in between you see some exoskeletons. Once you reach instar 5, they're getting close to diapause. Here we see one larvae still active, although the cohorts are hidden in the paper. We remove the larvae that are still feeding so that we do not have extra frass in the container. They are separated in a separate container and given fresh leaves. Here you see the cohorts in diapause between the sheets of paper. We cut away the packet where the animals are dormant and put it into a clean cup. They may get active again as we move them, but they will soon find a place to web up and remain that way for the winter diapause. We took some larvae and let them grow up in a terrarium. This is a natural environment, and as you can see, they're feeding on the various plants that are in the terrarium, preferring speedwell. They grew very well, shed from one instar to the other, and eventually we're looking for sites for diapausing. This is the instar 5 looking for a place for the winter sleep. We offer different materials for the larvae to choose a site for diapause, and common reed bundles were preferred. It can happen that the larvae become ill, in which case they do not develop properly and do not shed into the next instar. They are not responding and not curling up, which is a normal behavior when the larvae are healthy. They try to feed, but give up feeding and literally have no appetite. Another event that is not healthy is when the larvae has difficulty eclosing, that is, the butterfly to hatch from the larvae. This one has a lot of problems and in the end did not expand its wings. Although it is alive, it will not survive, and like this one, dies. The most important steps is to create labels for each of the groups of larvae as to the genetic background. These labels were moved from one cup to the other and stay with them throughout their development. Other information is entered on a daily basis into a diary. Beyond that, we fill out forms for various aspects of their development. The work of the Taylor's Checkerspot Conservation Breeding Facility came to fruition in April of 2015 when close to 300 larvae were released to the wild in the Denman Island Provincial Park Butterfly Reserve. Our mission is to raise these animals in captivity and find better ways for husbandry so to have them forever in the wild. Bye.